Hello, everyone. Yeah, cheers, and I want to start by saying Happy New Year to each and every one of us. We've not really been running our videos because we've been in the holiday mood, and I said, what better way to start the year than with a beautiful video on science and just something to kick off the new year with. So, I first say Happy New Year to each and every one of us. I pray that it is a wonderful year filled with love, laughter, free of illness. Now, today we're going to ask ourselves, what are vaccines and what do they do? What are vaccines? What are the types of vaccines and what do they do? Now, it's good to know that vaccines are what we call biological preparations, and they are prepared in such a way that they're expected to trigger or provoke an immune response within the individual to which they have been given to. So when you think of vaccines, you think of them as biological preparations that are prepared with the intention that they would provoke an immune response within the individual to which they have been given to. When you immunize children or you vaccinate people, the idea is that you're trying to provoke an immune response within that individual. Now, when it comes to vaccines, there are different types of vaccines, and we might ask ourselves, what are the different types of vaccines? Now, there are about 8 different types of vaccines, and we'll run through these 8 different types, try and see what it is that we can learn about these 8 different types of vaccines that we have, knowing now that vaccines are preparations, biological preparations that help to induce immune response. So, what are the types of vaccines? One, we have what we refer to as a live attenuated vaccine. Now, what's a live attenuated vaccine? This is a kind of vaccine in which the pathogen is still living. That's the pathogen that causes the illness is still living, is still very much alive, but it has been worked upon. So the key word there is attenuated. It has been weakened such that it is not able to, cause that disease with the intensity and virulence with which it will have caused the disease before if it was not weakened. So that's what we refer to as a live attenuated vaccine. And this kind of vaccine can be given in order to, elicit an immune response. There are different types of this kind of vaccine. We have the BCG that is used to treat tuberculosis. Bacillus calmette-guerin is a type of live attenuated vaccine. We also have other types of live attenuated vaccines. We have the yellow fever vaccine. It's a type of live attenuated vaccine. And then we have our MMR vaccine, the vaccine that we use for measles, mumps, and rubella. That's another type of vaccine, which is a live attenuated vaccine. Now, when we leave that, the next thing we have is what we refer to as an inactivated vaccine. An inactivated vaccine is a vaccine in which the pathogen has been killed, it has been destroyed by one or many means. You can use heat, so you can have a heat inactivated vaccine. That's a vaccine in which the pathogen has been killed and was killed using heat. Now, this kind of pathogen is also as effective. It may not be as effective as a live attenuated vaccine, but it's a bit safer than a live attenuated vaccine. And the pathogen is killed, and then the preparation is made, the biological preparation is made, and it can be given to an individual. So the first type is the live attenuated vaccine. The second type is the inactivated or killed vaccine, which the different types are the vaccine for the rabies virus, the vaccine for the intramuscular polio vaccine, and then we have the rabies vaccine. These are examples of inactivated vaccines. Live attenuated inactivated vaccines. Now we have subunit or recombinant vaccines. Subunit or recombinant vaccines. Now these are vaccines in which a portion or a part of the pathogen is taken and is used to make the vaccine. A portion or a part of the pathogen is taken, perhaps a protein, perhaps a monosaccharide from within the pathogen is taken and is used to make these vaccines. These vaccines can also be really effective, and examples are our pneumococcal vaccine, our meningococcal vaccines. These are examples of vaccines that are subunit or recombinant vaccines. So live attenuated, inactivated, subunit or recombinant vaccines. We also have what we call toxoid vaccines. And just as the name implies, toxoid comes from the word toxins. So vaccines that are made using the toxin that has been produced by the pathogen. It either be an endotoxin or an exotoxin. But once it's a toxin that has been, that is present within the pathogen, either as an exotoxin or an endotoxin. And this toxin is now harvested and now used to create a vaccine that can elicit, generate an immune response within an individual. This kind of vaccine, we refer to it as a toxoid vaccine. And this vaccine is important for us to know, and it's important for us to know its effectiveness. Examples of such vaccines are our cholera vaccines. There are examples of toxoid vaccines, our tetanus vaccines, our diphtheria vaccines. So DPT. These are examples of vaccines that are toxoid vaccines. Now we also have what we call mRNA toxins. That is messenger RNA toxins. So, messenger RNA vaccines, not toxins. Messenger RNA vaccines. Now these are vaccines in which, just as the word implies, it's easy to remember this. These are vaccines that are created by harvesting the messenger RNA component within pathogens. And when this component of pathogens, that's the messenger RNA that is within the component, within the pathogen, is harvested, it is used to make a vaccine. And this vaccine can be quite effective. Examples of these vaccines are our COVID-19 vaccines, the ones that were produced by Pfizer and Moderna. These are examples of mRNA vaccines. And then we also have examples of what we call a viral vector vaccine. The viral vector vaccines. Now, viral vector vaccines are unique vaccines because they are vaccines that make use of viral vectors. What are viral vectors? Viral vectors are viruses that have the ability, the capacity to be able to deposit particular genetic materials within a host cell. So if we make use of such viruses to, dispense or introduce genetic material into a host cell in order to elicit or generate an immune response, that kind of vaccine is called a viral vector vaccine. An example is the Ebola vaccine, the vaccine that is used against Ebola virus. Another example is the COVID vaccine, the one that was produced by Johnson and Johnson, is an example of a viral vector vaccine. And then we move from there to what we call a DNA vaccine. Now, within pathogens, we have plasmids. We've discussed plasmids a lot on this channel. We said they are extra chromosomal DNA. So if we make use of plasmids and use these extra chromosomal DNAs to produce vaccines, these kind of vaccines are referred to as DNA vaccines. An example of such vaccines are the vaccines that are produced against Zika viruses. These are examples of DNA vaccines. Lastly, we have what we call protein-based vaccines. Protein-based vaccines, and these are vaccines that what happens is that the protein component of a pathogen is taken, is harvested, and is used to make a vaccine. So we call that the protein-based vaccine. And an example of that is the Novavax COVID vaccine. This is an example of vaccine. So we've discussed what vaccines are: biological preparations, there to generate or elicit immune responses. And then we'll also discuss the 8 different types of vaccines and their examples. Do well to look through this video, pause it, and then listen to it again and again so that this can help you during your tests or exams to remember these things and put them down and be able to pass very well. Thank you for listening to this little discourse. Remember to like our channel. Remember to subscribe to our channel. Remember to tell your friends about our channel. And I pray that we'll have a wonderful 2025. Do have a wonderful day. Stay blessed.